Well, good evening, everybody. Good evening, Philadelphians, Phillies fans. Isn't it great that we organized this for 6 o'clock so that by 8:07, those of you who choose to be, it's a tough act to follow, but those of you who choose to be somewhere else, you'll be in good shape for that. Thank you for coming out this evening with all these things going on. I'm David Cole, President and CEO of the Science History Institute, and I'm delighted to welcome you to the 33rd Annual Ullyot Public Affairs Lecture featuring Dr. Carolyn Bertozzi. This annual award and lecture, endowed by chemist Glenn Ullyot, seeks to illustrate how chemistry, biology, and the sciences in general contribute to the public welfare. Glenn Ullyot said that, quote, "Chemistry, biology, and physics are the basic sciences that are key to understanding the world around us. It is the mission principle that each Ullyot lecturer will increasingly stimulate more people to appreciate the positive impact that these sciences and the researchers who pursue them have on our daily lives," end quote. I believe Glenn would have been pleased with this legacy as it continues to bring to the general public people of great scientific accomplishment who have changed and benefited the world. Among past Ullyot lecturers are Robert Langer, Philip Sharp, Jennifer Doudna, Roald Hoffman, and Francis Arnold. And it is our pleasure this evening to add Dr. Carolyn Bertozzi to this august roster. This annual lecture is particularly important to the Science History Institute as it allows the giants of science and medicine to bring their stories to a new audience. Stories of successes, failures, trials and tribulations at the bench and in the larger world. Their stories illuminate and contextualize the science all around us. And Science History Institute is committed to sharing these stories with a growing global audience of the scientifically curious. This evening's Ullyot Public Affairs lecture would not be possible without the generous support of the Institute's sponsors and partners, including the Department of Chemistry at the University of Pennsylvania, the Department of Chemistry and Biochemistry at the University of the Sciences, and the Philadelphia section and the Delaware section of the American Chemical Society. Thank you all very much. This evening, we and our sponsors and partners are very happy to welcome Nobel laureate Carolyn Bertozzi to Philadelphia. Following her talk, there will be a question and answer period, followed by our award presentation. During Q&A, if you have a question, please raise your hand and one of my colleagues in the room, and I think there are 3, will find you with a microphone. And now let me tell you just a bit about our distinguished speaker. Dr. Bertozzi completed her undergraduate degree in chemistry at Harvard University and her PhD at the University of California at Berkeley, focusing on the chemical synthesis of oligosaccharide analogs. She is currently the Baker Family Director of Stanford ChemH, the Ann T. and Robert M. Bass Professor in the School of Humanities and Sciences, and Professor, by courtesy, of Chemical and Systems Biology and of Radiology at Stanford University. Her research interests span the disciplines of chemistry and biology, with an emphasis on studies of cell surface sugars that are important to human health and disease. The Bertozzi Group develops chemical tools to study the glycobiology underlying diseases such as cancer, inflammation, tuberculosis, and most recently, COVID-19. She is the inventor of bioorthogonal chemistry, a class of chemical reactions compatible with living systems that enable molecular imaging and drug targeting. Her group also developed new therapeutic modalities for targeted degradation of extracellular biomolecules. Named a MacArthur Fellow in 1999, Dr. Bertozzi has received many awards for her dedication to chemistry and for training a new generation of scientists fluent in both chemistry and biology. She has been elected to the Institute of Medicine, the National Academy of Sciences, and the American Academy of Arts and Sciences. And she has received the Lemelson MIT Prize, the Heinrich Weiland Prize, the ACS Award in Pure Chemistry, and the Chemistry of the Future Solvay Prize, among many other honors. And last year, as you're well aware, Dr. Bertozzi was awarded the Nobel Prize in Chemistry for her development of bioorthogonal reactions, which allow scientists to explore cells and track biological processes without disrupting the normal chemistry of those cells. Several of the technologies developed in Dr. Bertozzi's lab have been adapted for commercial use, and she is actively engaged with several biotechnology startups. Dr. Bertozzi co-founded Redwood Bioscience, Enable Biosciences, Pallian Pharmaceuticals, Interven Bio, Ullyot Bio, Grace Science LLC, and Lycia Therapeutics. She is also a member of the board of directors of Eli Lilly. And so we are privileged to have in our midst this evening a path-breaking scientist, an esteemed teacher, an engaged leader, and a prolific entrepreneur. It is my great honor, therefore, to present our 2023 Ullyot Public Affairs Lecturer, Dr. Carolyn Bertozzi.
Thank you. It's a tremendous honor to be here in this capacity. Thank you, everyone, for coming out tonight. I grew up in Boston, so I understand that when things conflict with sports, you have a real problem, you know. And so I appreciate that you would take the time to come here tonight and share some science with me in a timely manner so you can make it to the Phillies game when they start. I thought what I would do is just tell you a story about some recent work from my lab. And by that, I mean about maybe the project that started around 15 years ago. I will make very brief mention in this talk about the work that was recognized by the Nobel Committee, which is even older than that, which tends to be the way it goes with Nobel Prizes. But hopefully the take-home messages will be clear, and it will have to do with the importance of sugar molecules in our bodies, opportunities to treat diseases by understanding what sugars are doing and how we can manipulate them, and how having a little bit of bioorthogonal chemistry, as we call it, can be really empowering for these kinds of studies. Okay, so let me start with the very first way in which I was presented with the field of glycobiology. And it happened when I was an undergraduate back, I'm going to say around 1985. And I was taking a cell biology class. And in that class, we were learning about the major biopolymers, as they're called. So these are the rather large molecules of biology. And so we learned about DNA, and we learned about RNA. And you've all heard a lot about RNA over the last 2 weeks here at Penn anyways. And we learned about proteins. And then finally, we learned about complex carbohydrates. And nowadays, we tend to call them glycans. But all our professor had to say about them at that time was that they form a coating on the surface of our cells. And in that regard, a cell is like a peanut M&M. And that was pretty much the level of detail in which we learned about cell surface sugars at Harvard University. What do you expect, right? I mean, you know. So, and our professor said, when you bite through a peanut M&M in particular and look at it in cross-section, well, that's kind of like a picture of a cell where there's a sugar coating on the outside, and then there's all this other stuff on the inside, right? You could picture the chocolate layer being like the plasma membrane, like the lipids and the fat. And then the nut is like the proteins and all the other stuff inside the cell. And when someone asked why are cells coated in sugars, our professor responded with something that was kind of right out of that famous commercial from the 1980s on peanut M&Ms, which is that because of that candy coating, they melt in your where do they melt? In your mouth, but not in your hands, right? Okay. In other words, the assumption back then was that the sugar coating on the cell served a protective function, which might have made it seem not particularly that interesting at that time. And that's all I knew until about a decade later, when now I was a graduate student doing my PhD and studying the chemistry of sugars. And by then, biologists had learned a lot more about what they might be doing on the surfaces of our cells. And now the metaphors had shifted, and people were no longer talking about cell surface sugar coatings like a peanut M&M. Now they were talking about those sugars as being more like the surface of the planet Earth, where if you flew around in a helicopter and kind of looked down, you would see all of these features on the surface of our planet: tall trees and short trees and shrubs and flowers and open fields. And a lot of it is in motion, right? The wind is blowing and things are moving. And now we think that's probably what the surface of a cell actually looks like, where the vegetation, the trees and the grass and the shrubs, are made up of these complex sugars. They're long chains, sometimes they're branched chains, and they have information. The structures of these sugars are trying to tell us something, and they're actually engaged in biology. So it became much more interesting to me to study sugars through this metaphor than through the peanut M&M metaphor. And pretty much every day since I first discovered these molecules in graduate school, I have worked on them in some way or another. So we now think of the structures of cell surface sugars. They're like a language. There's data embedded in these structures, and they affect how the cell behaves, and they affect how other cells interact with the cells in your body. They're trying to tell us something. And one of the things these sugars are trying to tell us is whether the cell that they populate is a healthy cell or a diseased cell. And that revelation made them even more interesting to me, who, as a scientist, is trying to improve human health through combining chemistry and biology. So let me just tell you something about sugars that you might not have known, but which has always been very important in your life. One of the things your cell surface sugars are telling us is your blood group. So we all have a blood group. Some of us hopefully know the blood group. That like to think that that's the preponderance of people in the audience, but we're about to take a poll. How many people in the audience have a blood type of O? Me too. Okay. So what all of us share in common is our blood cells are coated with a sugar that has this particular structure. And this is an actual chemical structure. And you can see there's different colors. Each color is a different building block. And the building blocks get stitched together in certain patterns to make these complex carbohydrates. So the black building block, the green building block, and the blue building block, when put together in this particular arrangement, is blood type O. Now, how many people have blood type A? Oh, a lot of people here in the audience. That's good for you. What you have, okay, what you have that the O people don't have is you have an enzyme in your cells that will add this red building block so that it's the same underlying structure here. These colors are the same, but now there's one more extra color. Okay, that's blood type A. And blood type B, let's have the Bs here in the audience. Usually there's fewer of them, and there's some, but not too many. You have a slightly different enzyme, and instead of adding the red sugar, your enzyme adds another one of these green sugars to make the blood type B. And then there's the rare people among us that might be AB. Yeah, proud, nothing to be ashamed of. Great. So you got one copy of this enzyme from one parent and a copy of this enzyme from the other parent. So you have kind of 50-50 of A and B, so you're AB. And this was a very important discovery in human medicine, because now that we understand the different blood types, we can figure out how to match a blood donor with a blood recipient. And if you make a mistake in that match, you can injure a person severely. So understanding the blood types and the structures of the sugars is what enabled what I think of as one of the most important procedures in all of human medicine, which is the blood transfusion. Okay, so that was one of the early discoveries in the world of glycobiology that kind of put this field on the map. But for me, I became fascinated with cell surface sugars when I learned how important they are in cancer. So it's been known since back in the 1960s, even before I was born, that if you look at the pattern of these sugars on healthy cells versus cells of a tumor that arise in a tissue environment, you'll find pretty dramatic changes in the patterns of the sugars. And there's one sugar in particular which undergoes a really big change. It's one of these building blocks. And so, by the way, these cartoons with the different shapes, those denote the structures of complex sugars, where each of these different shapes is one of those building blocks. So this is the biologist vernacular of complex carbohydrates. And the pink diamond is always used to refer to this particular chemical structure, which has a name, and we call it sialic acid. So that's one of the building blocks that are used to build the complex sugars. And our normal healthy cells have what I like to think of as a well-manicured garden of these sialic acids. So there's always some there, okay, but it's nicely controlled and landscaped. And by contrast, if you look at the pattern of the sialic acids on cancer cells within a tumor, it's as if that well-manicured garden overgrew into a tropical jungle. Like nobody has mowed the lawn or pruned the trees in a century. That's what it looks like. And that was first observed in the 1960s. And oncologists and pathologists commented on these observations again and again and again. But for more than half a century since the observation was made, nobody had a really reasonable explanation as to why this might happen. It was just an observation. So my lab started thinking about this back around maybe 2010 or so. And we had an idea for why that might be happening, which came to us by virtue of some discoveries that had taken place in the field of cancer immunology. So let's talk for a minute about the important role that our immune system plays in protecting us from cancer. So your immune system is made up of white blood cells called immune cells that go around your body, patrolling your body for signs of infection or signs of damage. And the way these immune cells, which are the green balls here, the way that they know whether a cell is damaged is they will form a contact with that cell. We call it a synapse. It's as if the immune cells are docking onto that cell and sampling the merchandise. And they're looking for signs of damage. And if they see those signs of damage, they will get rid of that cell. So here's a simplified view of what's happening at that synapse. Here's the immune cell. Here's the cancer cell. And there are molecules on the surface of the immune cell. They're proteins. They're receptors, which means that they're proteins that are going to look for a ligand or something to attach to on the cancer cell. And there's two flavors of these receptors. Some of them are called activating receptors. So that would be the bunch that are in green. They are looking for ligands on the cancer cell, which would be an indicator that there's some damage going on. And if enough of these activating receptors can interact with enough of the damage signals, then there's a consequence. Those activating receptors send a signal to the immune cell that this is a bad guy. We should get rid of this cell. And the immune cell launches an attack. It drops a bomb and blows up the cell and often calls for help. So other immune cells can come in and drop their own bombs on the neighboring cells. Now, at the same time, the immune cell also has another somewhat opposite type of receptor called the inhibitory receptor. And sometimes people call these inhibitory receptors immune checkpoint receptors. They do the opposite. These receptors are looking for molecules on the cell that would be an indicator of good health. And this is important because immune cells have a pretty tricky job to do. They have to tell the difference between a damaged human cell and a healthy human cell, but they're both cells of your body. And if the immune cell makes a mistake and accidentally kills a healthy cell, well, that's the beginning of an autoimmune disease. That's what's happening in your body when you have autoimmunity. Your immune cells are attacking your healthy tissue. So the checkpoint receptors are meant to mitigate that risk. And at the end of the day, if there's enough of these inhibitory receptors binding the healthy signals, that inhibitory pathway will override the activating pathway, and the immune cell will just kind of go to sleep and walk away and mind its own business. And ultimately, it's the integration of the signals coming through these two types of receptors that that immune cell has to look at and make a decision. Do I drop a bomb or do I go to sleep? Okay. Now, understanding this biology is what led to, I think, the biggest revolution in the treatment of cancer in my lifetime, probably, which is the development of immune therapies. And what the first generation immune therapies do is they block the interaction between inhibitory receptors and the healthy signals. And by blocking this receptor ligand interaction, they allow the activating receptor interactions to dominate the behavior of the immune cell. So sometimes we call these medicines immune checkpoint inhibitors or checkpoint inhibitor therapies. So why were people so excited about these new therapies? Well, back around maybe 2012, 2013 or so it's now been 10 years, the very first waves of clinical data were coming out where people were testing these immune therapies in human cancer patients. And here's an example of one such data set where two different immune therapies were tested individually. They were drugs that bind checkpoint receptors called PD-1 or CTLA-4. Those are the technical terms. And these are receptors on a type of immune cell called the T cell. And there was another group of patients which had the combination of the two medicines. And these were patients with advanced metastatic melanoma. Now, prior to these checkpoint inhibitor therapies, that was 100% fatal diagnosis. Nobody survived who had advanced melanoma. It's a very aggressive cancer. But when the patients were treated with these medicines, you can see that this is what's called a Kaplan-Meier curve. It's a survival curve over time for the patients. So, as a function of months, all the way out here to 48 months, so that's, 4 years or so, the patients were taking these therapies, and some patients didn't survive. You can see that there's a drop here in the number of survivors. But at a certain point, somewhere between 30 and 50% of those patients were able to survive beyond 4 years, and some of them were actually cured. And the word cure had never been used in a sentence with metastatic melanoma. Okay, so this was transformative, the fact that you could actually keep patients alive with these medicines. So lots of medicines were made once this was figured out. All of these medicines are very similar in the type of molecule. They're all called monoclonal antibodies. They're proteins that have to be administered through infusion, through an IV. And the very first wave of these medicines came from Merck through what's a drug now called Keytruda. Keytruda is Merck's biggest blockbuster in a long time. Bristol Myers Squibb has two, Yervoy and Opdivo. And after these three transformed the field, everybody else jumped on board. And now there's a dozen or so of these checkpoint inhibitor therapies that people can take for different kinds of cancer. Now, you probably know this already because I think the revolution was all over the news. Back in 2013, Science magazine gave cancer immunotherapy the title of Breakthrough of the Year. Nature did spotlights on these new drugs. If you were in the supermarket checkout line, you could have read about immune therapies in Newsweek or in Time magazine. New Scientist went so far as to speculate that immune therapy might be cancer's penicillin moment. I mean, that's a pretty bold thing to say when you think about what penicillin did for bacterial infectious diseases. Famous people were cured of their cancers. Jimmy Carter had metastatic melanoma in his brain. He was one of the first patients to go on Keytruda, and he was lucky in that it worked for him. And here he's still with us now. I have to check every day, but yeah, he's 98 years old or something, right? And the scientists who did the foundational work for this field, Jim Allison and Tasuku Honjo, received the Nobel Prize in physiology or medicine back in 2018. So this really was an amazing moment for the field of oncology, but also somewhat sobering because they really did not know which patients were going to benefit and which were not. And still to this day, we're not very good at predicting this. So if you are one of the lucky 50% who benefited, that's great. You know, you'll live hopefully a rich life. But half the patients had no benefit from these medicines, and no one really understood why. Here's some of the numbers. Melanoma, still to this day, is the type of cancer that is most responsive to the immune therapies. About 45% of patients sort of integrated over many trials can benefit. But these other types of cancers for which you can get a prescription for an immune therapy have much less favorable outcomes. You know, only 10% or so of patients might benefit. And we don't know why. So the big question in the field for the last decade has been, why doesn't everybody benefit from these immune therapies? And for the patients who don't benefit, are there some other mechanisms at play that are affecting the immune cells besides those checkpoint receptors and their ligands? Which brings me back to sialic acids. So here we have an observation that dates back more than half a century, that the cancer cells have overproduced complex carbohydrates with sialic acid compared to normal healthy cells. And guess what? It turns out those sialic acids can also put immune cells to sleep. And they do it by interacting with receptors on immune cells that are called the sialic acid binding immunoglobulin-like lectins. And that's a big mouthful with a lot of words. So we just abbreviate these receptors, the Sig-Lec family members, Sig-Lecs. There are 14 members of this family in humans. In this cartoon, this yellow line you can think of as the membrane or the surface of the immune cell. All of these little shapes that are in blue are facing the outside of the cell. And these bricks in red and purple are on the inside of the cell. Different members of the Sig-Lec family are found on various different types of immune cells. And this is another interesting feature of our immune system. I like to think of our immune system as like a military industrial complex, right? Where just as with the American military, we have the Marines, there's the Army, there's the Air Force, there's the Navy and the Coast Guard and the Navy SEALs, and everyone has their job. And the same is true in your immune system. there's different kinds of immune cells, and they all have their job, and here's a bunch of them that are listed down here. But regardless of their job, they all have members of this receptor family decorating the surface of the immune cell. Now, these receptors have parts outside the cell that bind sialic acid, and it's like this crescent moon is actually the sialic acid binding part. And every member of the sialic family has a crescent moon. Many members of the sialic family have inside the cell red and purple bricks that have these acronyms called the ITIM and ITSM motif. Those are immunology acronyms, and all you really need to know is that those are the exact same bits and pieces that are found in one of those checkpoint receptors that's the target of the first generation immune therapy drugs. It's the T cell checkpoint receptor called PD-1. So the way that a scientist would refer to these receptors is that they share a common signaling machinery. In other words, it's these red and purple bricks that allow these receptors to put the immune cell to sleep and shut it off and turn it down. And so both the Siglecs and PD-1, they do the same thing to the immune cell, but on the outside of the cell, they're binding to different kinds of molecules. And the first generation of immune therapies blocks PD-1, but has no effect on any of the Siglecs. And look how many Siglecs there are compared to PD-1. So we started working on this problem with the hypothesis that the T cell with its checkpoint receptor binding to a ligand on the cancer cell, that this is just one pathway by which a cancer can put the immune cell to sleep. But meanwhile, there's all these different Siglecs binding those carbohydrates with sialic acids on the cancer cell. And there's so many of these Siglecs that immune cells can go to sleep anyways if you block this, as long as they have these interactions. And so we therefore speculated that maybe a more effective immune therapy would be one that blocks this interaction, or that maybe combines a blocker of the Siglecs and their ligands with a blocker of PD-1 and its ligands. That was the hypothesis. So we started working on this, and we convinced ourselves that yes, the reason cancers have so much of these sialic acid glycans is because that allows them to trick immune cells and hypnotize them. And if an immune cell, if a cancer cell can trick and hypnotize an immune cell, that cancer cell lives another day and proliferates and grows and spreads. So here's the idea, okay, in a nutshell. Immune cells, when things are working well for you, are able to sample signals on the cancer cell that give it a bad taste. And that bad taste causes the launching of a bomb. But if that cancer cell happens to have a lot of sialic acid, if the garden has grown into a jungle, now Siglec receptors can engage those sugars, and that tells the immune cell, hey, this is fine, this tastes delicious, okay? Nothing to see here, move along. Anybody see Star Wars, first one, 1977? Okay, it's a Jedi mind trick. All right. So now, understanding this biology, how do you make a therapeutic that messes with the good taste on the cancer cell? Well, we thought long and hard about this, and our first idea was just to take a page from the playbook of all of those pharma companies that made monoclonal antibodies that block the checkpoint receptor on the immune cell. So maybe one could make such a protein that binds to a Siglec and plugs it up. But the problem is there's so many Siglecs. There's not just one, there's 14. And do you have to block them all? You would have to make 14 different medicines. Or do you have to block 2 or 3, and which 2 or 3? And we didn't know. And so absent some knowledge about picking one or a few Siglecs, we thought maybe this wasn't such a great idea after all. Now, by contrast, one thing we did know about all the Siglecs is that they need to bind ligands that have the sugar sialic acid. That's something they all share in common. When they bind sugars, there has to be a sialic acid there. So we thought maybe we should target the sialic acid on the cancer cell with a drug rather than trying to plug the Siglex. So that's exactly what we did. And back to the vegetation metaphors, we thought, wouldn't it be great to make a drug that acts like a molecular lawnmower? So imagine parking this drug on the cancer cell, turn on the motor, right? And just let it mow the lawn, right? Get that garden into shape, take off those sialic acids. So that's what we did. And the form that that lawnmower took at the level of the molecule was a fusion between a cancer binding antibody, which is this part here in pink. So this is exactly a monoclonal antibody, like so many that are known to be drugs today. And we fused that cancer binding antibody to an enzyme called a sialidase. And sialidases cut the sialic acid sugars off of whatever lies beneath them. And those are enzymes that are known in the world. So our first prototype molecule was made with an antibody by the name trastuzumab. It's already an approved drug that binds to a protein called HER2, that's HER2, which is overly abundant on breast cancers. So it's a breast cancer medicine that's right now used, and you can get this drug from Roche and Genentech. They made it together. And the brand name of the drug is called Herceptin. This is the active ingredient in Herceptin. And so this antibody will park on the breast cancer cell because those cells have the HER2 protein. And then for the sialidase, for the prototype drug, we chose an enzyme from a bacterium. It was actually from Salmonella, of all things. And that was just a practical choice. It was an easy enzyme to work with in the laboratory. Now, we had to figure out how to connect this protein to this protein. And there's a number of ways a person can do that. But as a chemist, we thought maybe we could use our chemistry skills to actually form covalent bonds between these two proteins. And so this is a good excuse for me to introduce the concept of bioorthogonal chemistry. This is the only chemistry slide, by the way, in the presentation. But this is why I got the Nobel Prize. Okay, so you might as well learn it. Oh, yeah, okay, thanks. All right, so terminology. Bioorthogonal means not interacting with biology. And that is literally what's so special about this chemistry. These are chemical reactions that you can perform between two different chemical groups. And it doesn't matter if those chemical groups are surrounded by all of the mess of biology. None of that mess will interfere with the chemistry because it's so precise and so selective, okay? So we used two different bioorthogonal chemistries to connect the two proteins together with a bond. The first of these chemistries we called the Pictet-Spengler ligation. And by the way, I should tell you that one of the themes of bioorthogonal chemistry is that we go back 100 years to the literature of the previous century, much of which came out of Germany pre-World War II. And there's some beautiful pearls of chemistry in that literature. And then we repurpose those chemistries for the modern application in biology. So Pictet-Spengler ligation was first reported in 1905. Okay, but we kind of repurposed it so it would work with our proteins. And what we do is we produce the antibody with aldehyde functional groups positioned exactly where we want them. And we have a technology for that. Then we chemically react the antibody aldehydes with a molecule like this one, where this dimethyl hydrazine indole, for the chemists in the audience, will react first through an aminium-ion intermediate, then there's an intramolecular electrophilic aromatic substitution. Are you with me, everybody? And then you form a carbon-carbon bond, and then you've got this piece on the antibody. Now, in parallel, we take the enzyme, and we do a little chemistry on the enzyme to put this piece, which has a group here, and you might notice there's an 8-membered ring with a triple bond in the ring. That's called a cyclooctine. The cyclooctine will very precisely react with this group, which we added to the antibody. It's really small on the screen here, but it's 3 nitrogen atoms. That's an azide. So the azide and the cyclooctine react in another bioorthogonal reaction called copper-free click chemistry, so named because it's very similar to another chemistry called click chemistry with copper, developed by Barry Sharpless and Morton Meldal, who shared the Nobel Prize with me, okay? So this is how we chemically conjugated the enzymes to the antibody, okay? So now, does the antibody sialidase conjugate shrink tumors? Well, we go into mouse models to test that initially. And this is a model we ran with my good friend Heinz Laubli at University Hospital Basel and his former grad student, Michael Stanzak. It's a model that they set up, which emulates human breast cancers that are normally treated with the antibody, trastuzumab. The way that Michael did this is he took a mouse breast cancer cell line called EMT6, and he engineered that mouse breast cancer so that it would present the human HER2 on the surface, just like a human breast cancer would. Then he injected those cancer cells into the mammary fat pad. So the mouse has a breast cancer in the breast tissue. And at time 0, the mice get injected with the tumor cells, and then the tumors start to grow and grow and grow, and you can monitor their growth over time. Now, if you treat these mice either with a saline solution as a control, or with the antibody just naked, alone, with no modifications, the tumors grow at the same rate. That's because it was known this model is resistant to trastuzumab, all right? But when we put a sialidase on the trastuzumab, that's what T-sia is, trastuzumab sialidase, at 2 different doses, you can see now the tumors have a really hard time growing. And when Michael harvested these little lame little tumors, they were just infiltrated with immune cells, and the immune cells were killing the cancer cells. So this gave us some encouragement that maybe we had a pretty good idea for a new kind of immune therapy. So at that point, we wanted to make antibody sialidase conjugates that were more amenable to drug development and human clinical testing. And the best way to do that, if you're an academic, is to start a company and raise enough money to actually do the translational science. So I co-founded a company called Palion Pharmaceuticals. They're located in the Boston area. And the scientists at Palion went to work to try to basically improve upon the design and make a molecule that was more developable, is the word that people in the industry would use for a human medicine. So the changes that they made are, first of all, we had used a bacterial sialidase, but that's not ideal for a human medicine because a foreign protein in our bodies can elicit an immune response against the drug, and that's not good. So they swapped out that bacterial sialidase for one of our own human sialidases. That was a big improvement right there. You can see they changed the shape and the architecture. So we had put 2 enzymes on our antibody, but that made it a really big molecule. And they shrunk it down by just taking one of these arms off the antibody and replacing it with the sialidase. So the overall weight of this molecule is similar to a natural monoclonal antibody. And then finally, whereas we had made a chemical fusion using our bioorthogonal chemistries, The scientists at Palion thought it would be easier to manufacture this medicine on a large scale if they could make it through what's called a genetic fusion. So much to my chagrin, there is no bioorthogonal chemistry in the actual clinical candidate, okay? And this new molecule, which they called the eagle, we're in Philadelphia, I had to share that with you, okay? And you can kind of see why, if this is the head and that's the beak and here's the wings, it kind of looks like an eagle. And they tested that eagle in the same animal model, that HER2 positive mouse breast cancer model. So these are data from Palion. So here's the eagle being tested. Each of these curves is tumor growing in a different mouse. So there's 6 different mice here, 6 different curves. And when the mice were treated with the eagle, which is in blue, you can see that the tumors, they started to grow, but then they kind of petered out. And it was only in a few different animals where the tumors were able to take off, okay? So we call that 3 out of 6 mice had a complete response, okay? The others had a partial response. And in the same experiment, different mice were treated with the existing checkpoint inhibitor, antibody against PD-1, which was known to be a good drug for this animal model, even though it isn't for some patients. And sure enough, as was reported previously, about 4 out of 6 mice had a complete response. But when we combined these two together, we cured every single mouse of their breast cancer. And these mice had developed immunological memory. So once the immune system had cleared out the cancers, good luck trying to grow new cancers. These mice had been vaccinated against these tumors. So right now, Palion has 1 eagle in a human clinical trial. It just finished a phase 1 study, and they're enrolling in a phase 2 study. And they have another eagle that's going into the clinic probably early 2024. So all of our fingers are collectively crossed that we can actually bring some benefit to patients with these lawnmowers from glycoscience. Okay. So what are the take-home messages for you? Okay, first of all, if you remember nothing else, remember that all cells are coated with sugars, and they're not just there to keep the cells from melting in your hand, okay? It's more complicated than that. In fact, cancer cells have altered sugar patterns, and those altered sugars help them survive and grow and spread by preventing immune cells from killing them. So that was a revelation of the last 15 years or so. In particular, cancer cell surface sialic acids are binding to those sialic receptors on immune cells. And that interaction helps them escape the surveillance of the immune system. So if you've absorbed bullet point number 2, you can move on to some of the technical details of bullet point number 3. And then finally, we made a new kind of medicine. It's a fusion between an antibody and a sialidase enzyme that parks on the cancer cell and mows the lawn and gets those sugars off so that immune cells can do their job and stay awake while they're doing it. Okay, so with that, I always like to thank my research group. Even though today's talk was at a very high level, covered a lot of work over more than a decade, there are people behind the scenes doing all of this work, working really hard. And so I'm the mouthpiece, but they are the hands and the heart and the soul. And we can't do any work unless people support it financially. So these are the organizations that have funded my research. But most importantly, thank you for coming out tonight, showing your support and enthusiasm for science. And I'm happy to take questions. Thank you.
Stuff of which Nobel Prizes are made. That was incredibly stimulating and refreshingly clear. Thank you so much. A master class. Yes, doubtless there are questions. My colleagues are here to help you with microphones to take questions. Take it away.
Microphone has to be turned on.
Hi, I have a quick question. If what you do is mowing those cells, will it be beneficial to regularly mow people's cell, and so the immune system will keep attacking the ones that could become cancer cells?
Okay, so I think the question is, how do we prevent the medicine from mowing the wrong lawn? Yeah. Oh, how do we keep the mowing going? Yeah, okay. Yeah, that's a good question. So two thoughts on that question. One, when we put the medicine into the patient's bodies, it will stay in circulation, coming in and out of the tumor, for about a week. Okay, so for a week it's in there and it's mowing. Once the drug is cleared out, the clock is now ticking, and eventually, like your lawn, you mow the lawn, the grass will grow again. The grass starts to grow, okay? So one thing that we did in our phase 1 clinical study is ask the question, how fast does the grass come back versus, the medicine staying in circulation? And this is important because it tells us how frequently do we have to dose to keep the grass nicely mowed, okay? And from all of that, we came up with this dosing schedule of once a week. We would like that dosing schedule to be less frequent, but we have to make some improvements to the molecule to have it last longer in the body. And might you have the microphone?
Use microphone, thanks.
But like, would it be beneficial for a healthy person to take it so the mold will continue? That'll be my last question.
All cancer medicines, all medicines, period, have costs and benefits, right? The safer the medicine is, right, the longer a person can take that medicine, generally speaking. So we hope that our medicines are very safe and very well tolerated. And in the phase 1 clinical trial, we had no significant adverse events, but those were fairly short-term trials. These are trials of just a few months. For other immune therapies, the major side effects, as you might predict, are autoimmune side effects. So people who take these first-generation checkpoint inhibitors often end up developing autoimmune diseases after they've been on the drug for a year, let's say. And at some point, the autoimmune disease might be so severe that they have to stop taking their cancer medicine. We'll see what happens. Our drug is very early in the clinical testing, so we don't know yet what the longer-term side effects might be. But if a patient is able to stay on the medicine without significant autoimmune side effects, then usually the longer they stay on, the better, because you're more likely to keep a cancer at bay. But I don't think that we know yet what that's going to look like.
Couple up front here.
Oh, thank you. I have a question. With the therapy, is it concentration driven, or you mentioned orthogonality? Is there a spatial component to the way the molecules are interacting on the cell surface in addition to just, say, a coverage standpoint?
It's a good question. I think maybe what you're asking is, does it matter like whether this is here or here or here, or what's the space between these things, those kinds of parameters?
Yes.
Yeah. Early on in this project, we thought it would matter. In fact, the reason of all places to put the green enzyme, we put them down here because we wanted to keep them away from these parts, which bind the antigen. And also we wanted to keep them away from some parts in the middle here, which are involved in immune effector functions. And we actually made a bunch of different constructs where we moved these enzymes around in different places. But most of these constructs behaved similarly. It didn't really matter so much what the specifics of the geometries were. And then when we dug a little deeper, we discovered that once the antibody parks on the cancer cell by binding to HER2, it's actually very mobile. So it's driving really fast on the cell surface and just mowing very quickly. So, within in less than an hour, these cells were stripped entirely of their sialic acids from just all the driving, right? So it might just be that, again, there's so much motion and dynamics that the specific geometries are just not as important as we thought they might be, which is very helpful, actually.
Dr. Bertozzi, do you think that the technology of using the lawnmower enzyme for sialic acid residues on cells might be helpful in the treatment of Alzheimer's disease?
Oh, what a great question. Possibly, and the reason I say that is going back to the Siglex. I showed you the cartoon here. Some of the sialic receptors are found on immune cells in the brain called microglia. And that's true for CD33. This is sialic 3, that's on microglia. And then this one, CD22, comes up as we age on microglia. And what they do on microglia is prevent those immune cells from eating stuff. And microglia are there in your brain because you have toxic stuff that builds up and it needs to get eaten and digested. And amyloid plaques are examples of toxic proteins that need to get eaten and digested and cleared away. So the sialics on the microglia prevent them from eating aggressively. And there are some data suggesting that if you block the function of those microglial sialics, those cells are now hungrier and they'll eat more. In fact, there's even a genetic mutation that's found in a significant minority of people where they have lost the function of sialic 3, and those people have a much lower incidence of Alzheimer's disease. So there's interest in targeting sialic 3 in particular for Alzheimer's disease, and there's at least one company doing that.
Hello, this is such a pleasure to be in this audience and to hear your presentation. My question is sialic a post-translational modification? And if so, is the growing of this grass or the weeds, is that because of a genomic influence or because of something happening post-translation?
This is a great question. I have a whole other talk that I will give on that. Not here, don't worry. Yeah, so the sialic acids are part of a bigger post-translational modification of cell surface proteins. This cartoon is probably better. You can see, here's an example of a protein. That's the long string in the backbone there. This is a protein anchored to the plasma membrane, decorated with sugars, and the sialic acid is at the cap of those sugars. So the entire complex carbohydrate is the post-translational modification, and the sialic acid is part of that structure. What happened inside the cell, right, that took it from the well-manicured garden to the tropical rainforest? We've done some work to try to figure out, like, what happened at the level of gene expression that led to a change in the sugar structures. And it turns out there's two kinds of changes that are happening. One type of change is there's an increase in certain enzymes that load the pink diamonds onto the underlying scaffold. Those enzymes are called sialyl transferases, and some of them are regulated by transcription factors known to be oncogenes. So how many people here have heard of the MYC oncogene? GSK people, get your hands up. So the MYC oncogene is a transcription factor that is overabundant in like more than half of human cancers, and it's kind of a root cause of a lot of cancerous biology. And that transcription factor activates the enzyme that puts pink diamonds on, okay? But also the underlying tree can be important as well. So there are certain proteins that carry tons and tons and tons of sugars. They're called mucins. And those mucins are also upregulated by certain oncogenes. So it's not just the pink diamonds, it's also the underlying scaffolds that present them. And they can both contribute.
Okay, thank you. It was so awesome. Also, if I could get a picture with you, perhaps afterwards, I would be so happy.
Happy to take pictures.
Question here, Jessica's here.
Hi, over here.
Oh.
Thank you for all your talk and your great work. It was extremely interesting. The question I have is, as far as I understand the talk, what the process, the engineering of it does is to identify and mow the grass to let the immune cell do its work,
Right.
properly. Was there any thought in trying to engineer a mechanism that would try to go after the root and do the work itself rather than have the immune system have to do anything?
It's a great, great question. So, in the world of oncology, we use every tool at our disposal, every modality at our disposal. And usually we end up combining drugs together because no single drug on its own is usually very good at killing cancer cells. The kind of medicine you're thinking about, again, back to the vegetation metaphor, you might think of it as a weed killer, right? If you can park a sialidase on a cancer cell and mow the grass, could you park some weed killer and inject it into the cell. And yes, you can. And there's a whole class of medicines. Let me get a structure here I can play with. There's a whole class of medicines called antibody drug conjugates. Let's get a picture here. So an antibody drug conjugate has the same kind of antibody, but then instead of these enzymes, there's a toxic molecule. And those medicines, they park on the cancer cell, and then they get in there with their toxic molecule, and then they kill the cell that way. And there's some very important medicines that are antibody drug conjugates. More than a dozen of them now have been FDA approved. Inevitably, the cancers in the people that are taking these medicines, they become resistant ultimately. And they learn how to spit the drug out. They learn how to deactivate the drug. So it's not enough just to have weed killers. It's really great if you can have immune therapies and weed killers, or to take patients that became resistant to the weed killer and now give them the immune therapy. And that's kind of how it's playing out in the clinic.
Wow, that's amazing. Thank
Yeah.
you.
I think we have time for two more questions. I know this hand has been up for a while here.
Hi, so I was wondering, for the trials of the eagle, the ones where some of the mice didn't have a complete response, it looked like for some of them, the tumor was growing and then it kind of stopped growing and then it went back up, and that was the non-complete response. I was wondering why that was the case, where it kind of had a period where it went down. Was that like the lawnmower like ran out of gas or?
Yeah, so it's interesting because these are measurements of tumor volume. And tumors can become more voluminous in two different ways. One way is that there's more tumor cells there. The cancer cells are growing, they're not dying, and they're getting bigger and bigger. But the tumor also grows when immune cells are infiltrating it and filling it up with other types of cells. And so in these experiments, after the treatments, which are the little arrows, at that point, there's a period of time where tumor cells are dying, but immune cells are also coming in and filling it up. And so the net balance looks, this kind of weird thing. Eventually though, like, here, when the tumors grow again, those tumors that's tumor, that's not immune cells. That's what's happening there.
I think we'll have our last question, and I'll just add that I know a few others have questions. What I'll say is that there is a reception, which I'll be saying a bit more about shortly, and perhaps you could approach Dr. Bertozzi at that reception and ask a few more questions, but I think this gentleman has the final word.
Thank you for that talk. The sialics, where you showed all the structures, you said there are too many of them to target with antibodies, but the crescent shape, how many different versions of the crescent shape are there?
Oh yeah, this is a really great sialic aficionado type of question. All the crescents are slightly different. They're not identical. And they're not redundant either. They have very discrete types of structures that they prefer to bind. They all need to have the sialic acid as this tip, but what distinguishes them is the stuff beneath it. So the sialic acid can be capping all kinds of different structures, and it's what's underneath that gives the specificity of the sialic for one type of sugar versus another. But at the end of the day, if you take away the sialic acid, the binding affinity plummets. So that is an Achilles heel for all of them.
Excellent questions, and let's thank Dr. Bertozzi one more time for a superb presentation. But that's not all. It is our tradition at the Science History Institute to honor our Ullyot lecturers with the presentation of the Philadelphia Liberty Bowl in recognition of their extraordinary accomplishments. Yeah. We present the Liberty Bowl to recognize extraordinary accomplishments in service to science and humanity. It is now my pleasure to invite to the stage Matt Irwin, Chair of the Philadelphia section of the American Chemical Society and Principal Investigator at DuPont, to present the Liberty Bowl to Dr. Bertozzi. Matt, come up, please.
All right, well, thank you again, Dr. Bertozzi, for a fantastic lecture. Really appreciate it. So now it's my honor as the chair-elect of the Philadelphia local section of the American Chemical Society and the co-chair of the Ullyot Scholarship and Lectureship Committee to present the Liberty Bowl to Dr. Bertozzi. As just noted, the Liberty Bowl is a handmade porcelain bowl. It has information about the Declaration of the Independence on the inside and interesting vignettes about Philadelphia tradition all along the outside. You'll see the Independence Hall and many other famous locations. So hopefully remember Philadelphia by. So thank you again, and I'll be very carefully hand this to you.
That sounds great. Officially presented. Commemorating the occasion, so. Congratulations.
Thank you.
We won't burden you with that at the reception. Well, I want to say that video of this evening's program, if you'd like to see it again, or you want to commend it to someone else's attention, will be posted on the Science History Institute's YouTube channel in the coming days. So we get to relive this terrific presentation. We want to encourage you to keep the conversation going by joining us in the Dow Public Square. That is our space up on the third floor of the building, for a reception in Dr. Bertozzi's honor. The Dow Public Square can be accessed via stairs and elevator right out here in the lobby. And if you wish to visit our museum as well, it will remain open until 8 p.m. In other words, just about time for first pitch. Thank you again, and I wish you all a very good evening. Thanks for being here.